right, and with that, I'm going to turn things over to Shahryar to introduce today's speakers. Shahryar, you may know, is the research leader for the USDA-ARS Cereal Disease Lab in St. Paul, Minnesota. He's also an adjunct professor in the Department of Plantology at the University of Minnesota and a leading expert on oat crown rust. Thank you very much, Shahryar, for being here.
Thank you, Jim. Thank you for the invitation to speak at the Speaking of Oats. I'd like to introduce you to two of our great scientists at the Cereal Disease Lab. There are many of them, but two of them are here today to tell you more about oat rust and crown rust, but also talk about rust in general and what CDL does. Dr. Oluseyi Fajolu, who is a biologist at the Cereal Disease Lab, and she'll tell you all about why she needs rust samples. And then Dr. Nazareno, who just graduated from the Department of Plant Pathology and now is a postdoctoral scientist, will tell you all about what he has done on oat crown rust and how that is of value to you. So Oluseyi, you're up.
All right, sorry about that. Hello, everyone, and thank you, Shahryar, for the introduction. And before you listen to the good, the bad, and the ugly, I will introduce to you briefly the Cereal Disease Laboratory and Rust Survey. I will also talk about the importance of collecting rust samples and share a few survey results. Our mission at the CDL is to reduce losses in cereal to rust and fusarium head blight. And here are the short description of rust labs at CDL. Shahryar's lab focuses on oat crown rust. And common to all the rust lab is a continual monitoring of rust population in the field. And this makes collection of rust samples crucial for our mission. So my message to you today is please send us rust samples. Why collect rust samples? This is highlighted in a recent rust disease brochure. Rust are among the most devastating cereal diseases, and unfortunately, those pathogens occur every year. The virulent isolate present on the field could vary from year to year, but the good news is the CDL is committed to research that help protect cereal from rust epidemic, but we need you. We need you to send us rust samples. We need you to share the information. We can send the rust brochure to you to distribute on your field days, as well as the sampling envelopes and the self-addressed stamp envelopes, all for free. The goal is to get more rust samples. The CDL coordinates annual rust survey. We scout the cereal fields for rust diseases, and observations gathered are submitted to the rust listserv or sent to my email. Most importantly, rust samples are collected and sent to the CDL. Please, we need those rust samples, and please collect and send rust samples to us. Information from the rust survey is summarized and published in the rust bulletin. This is also displayed on maps for our virtual distribution of rust pathogen across the US. Now let's talk about oats. On the screen is one of the results from our oat crown rust survey. The box plot is showing the virulence profile of isolates collected over 25 years on oat differential lines. On the y-axis is the number of virulence, that is, the number of resistant gene in oat crown rust, in oat differential lines that are broken down by oat crown rust isolates. On the x-axis is the year, and each box, it's a category of all isolates tested in that particular year. Each isolate was tested against 26 differential lines. For a better understanding of this data, let's review a box description. Starting from the minimum value to the maximum value. I will point your attention to three things on this graph. First, the minimum number of virulence in 1993 was 0. After 25 years, the minimum number of virulence had increased to 12. The mean number of virulence per isolate in 1993 was approximately 8. But in 2017, the average number of resistant gene defeated per isolate was 17. And thirdly, isolates collected in 2016 defeated 25 out of 26 differential lines tested. This result indicates isolates are becoming more virulent over time. To further illustrate the virulence profile of isolates is the heat map. On the y-axis are 40 differential oats lines. And those differential lines were tested with isolates collected in 1990 and isolates collected in 2015. Yellow means isolates were avirulent, while red means isolates were virulent to differential lines. Let's take a few seconds to think about the change of differential lines from yellow in 1990 to red in 2015. For example, PC91 gene was effective against all isolates tested in 1990, but not anymore. Similar to PC68 gene and a few other genes, it is obvious that the recent isolates have defeated most of the resistant genes in the oat line. Once again, I'll plead with us to please send us rust samples so that we can continue to monitor the virulence of rust population in the field. Remember, rust pathogens evolve in response to the resistant cultivar released on field. And with that, I want to say thank you for your attention, and I will turn it over to Eric.
Thank you, Oluseyi. So my presentation is entitled Oat Crown Rust, the Good, the Bad, and the Ugly. We all know that in each pathosystem, there's a good side and a bad side. And today we'll talk about one of the most important diseases of oat, which is oat crown rust. So let's start with the bad and ugly. Probably the ugliest or the worst aspect of this disease is that it causes 10 to 40% yield losses worldwide. Just in Minnesota and South Dakota alone, we suffered an estimated of $10 million per year losses. So this is a very devastating disease. And why is it very devastating? Is it because of the biology of the pathogen? So we can see here in the asexual phase, it reproduces very rapidly. And so the farmer doesn't realize that his field is already infected. And also in each step of the pathosystem, it can generate variability. It can generate variability through mutation and recombination, and also even somatic recombination. So that's why this disease is very hard to control. Oat crown rust also contributed to the world oat production decline in the last 60 years. Here we can see a drastic decline in oat production after the 1950s. Of course, the main factor was farm mechanization, which led to less use of oat as livestock feed, and also low prices compared to competitive crops like corn and soybean. And there's also some other biotic and abiotic stresses that contributed to this. Another bad thing probably is in the US, the oat importation exceeds production. So we can see in this graph, the US constitutes about 76% of the total world imports of oat from 1996 to 2020. And that amounts to 1.8 million tons per year, when we can only produce 930,000 tons. So there's a great opportunity here for us to help our US growers, particularly now that there's an increasing demand for oat for human consumption. So that's all for the bad things, really. Now we move to the good things. These are the recent advances in oat crown rust research in the past decade or so. So in 2013, the first oat consensus map was released. It was followed by the second oat consensus map in 2016, and that was saturated with more markers in 2018. And in 2019, the first diploid genome assemblies were released, and we saw last year and this year, the versions 1 and 2 of the OT3098 genomes were released, and including transcriptome assemblies. That's on the side of the host. On the side of the pathogen, the crown rust reference genomes were released in 2018, 2 of them. And then in 2020, the first population genomic studies and GWAS to identify effectors were also conducted. And a lot of things are also going on right now. We are building a pangenome of crown rust, and also a diploid level assembly of crown rust. And yeah, a lot of good things are happening on both sides of the pathosystem. But today, I'd like to talk about 3 projects that I worked on. Number 1 is the reference anchored linkage mapping for oat adult plant resistance. The second one is the marker assisted pyramiding of these APR loci. And the last one is the GWAS to detect putative effectors from crown rust. So starting with linkage mapping. So I just want to give a brief background on the 2 types of resistance that we use in breeding. Number 1 is race-specific resistance. We all know that this is the classic gene for gene resistance. So when we have the AVR protein from the pathogen, it gets detected by the R protein from the plant, and that induces effector-triggered immunity, and therefore there is resistance. But then when the pathogen evolves, it makes a different effector. That effector can no longer be recognized by the R protein, and there is no effector-triggered immunity, and that leads to susceptibility. And in oat, that happens in just 5 years or less. So it breaks down pretty easily. That's why we're looking at the other type of resistance, which is called adult plant resistance or partial resistance. It is deemed to be effective against a wide range of pathogens, and it has a different mechanism compared to the race-specific resistance. So here are some examples. We have LR34, LR67, and YR36. So as we can see, they are not NBS LRR genes. LR34 is an ABC transporter, LR67 is a hexose transporter, and YR36 is a kinase and a start domain protein. So they work differently compared to NBS LRR genes. Recognizing the value of the APR in breeding, Dr. Marty Carson screened different entries of oat in the mid-2000s. So what he did is he screened these oat accessions in a Bucktor nursery, and then he collected bulk uridinospores from the nursery and inoculated the seedlings in the greenhouse with the uridinospores. And then he collected only the virulent pustules from each entry, and later on inoculated each entry with their own virulent population. So that is all to eliminate the race-specific resistance and only select for adult plant resistance. So we can see in this graph, this is the mean severity in the inoculated nursery. This is the mean severity in the Bucktor nursery. And if the line falls along the regression line, if the oat entry falls in there, they can be suitable APR parents. So those that are in the boxes were selected for population development. Here are the 9 populations developed by Dr. Carson. I split them into 2 groups. The first group has these 5 populations, and the second group has these 4 populations. We all screened them in the Bucktor nursery because there are multiple races in there, which would enable screening for adult plant resistance. This is the mapping approach that I use for identifying QTL from the recombinant inbred line populations. So I phenotype them in the Bucktor nursery, send them for Illumina high-seq genotyping, and then call the reads against the OT3098 reference genome and call the SNPs, and those SNPs will be used for linkage mapping. This is just to illustrate the Cranner's assessment scale that I use in the field. So I collect 2 sets of data. The first is reaction class. So these are just from resistant to susceptible converted to numerical values. These are the representative pictures of these reaction classes. And here is the guide for scoring disease severity per entry. So I multiply disease severity by reaction class to get the coefficient of infection. This is the phenotype data for group 1 populations in the Bucktor nursery. Here we can see number of lines. This is disease severity, reaction class, and coefficient of infection. We can see in the coefficient of infection, these 2 populations are similar, and so these 3 populations. And so all these 5 populations were subjected to bulk segregant analysis. That means we select only the lines with extreme phenotypes. So up to 48 total lines were selected per population. And we did QTL mapping using that and found that these 2 populations actually have the same QTL, and also these 3 populations. But with more funding, we were able to send them for complete genotyping of all individuals in the population. So we selected one population from here, CI9416, and one population from this group, which is PI189733. And with the whole population genotyping, I conducted a simple interval mapping, and that validated the results of the box segregant analysis. And in here, we can see in this population, there's a QTL in for chromosome 40.1 named QPC-APR-40.1. And this in another population, we have this QPC-APR-40.2, which is different from 40.1. And we detected an additional QTL with a smaller effect in chromosome 6C. So here we can see LAD score, and the dashed line is the threshold for declaring QTL. This is just zoomed in version of the QTL that I've shown you in the previous slide. So this peak right here is for the simple interval mapping. I use another approach, which is composite interval mapping with marker cofactor selection, and that significantly decreased the interval. So as we can see here, that's the peak, and the effect is 43%. And I also computed for the linkage disequilibrium of those markers in that region, and found that there is high LD in those significant region, which is what we want for marker-assisted selection, because we don't want recombination within that QTL. We want the QTL to be inherited as one. So this is just showing you the relationship between the physical order and the genetic order. The same is true for this QTL. We narrowed it down, and the effect is 39%, still high LD in that significant region. And the markers that you see in bold have been converted to CASP or PACE markers for selection. The third QTL has an effect of 22%, still high LD, and we've developed markers for them for this QTL. And so we conclude here that BSA markers only identify the approximate location of the QTL, but with the reference genome, we had more resolution and more power. I was interested in looking at the gene content of the significant regions, and this is just one representative QTL and some statistics on the chromosome and significant regions based on the 3098 reference genome. And I found that, yes, indeed, there are genes that can control APR. In this significant region, we can see kinases, disease resistance protein, and several other genes that can confer resistance. So we need fine mapping to really pinpoint which gene is causing the phenotype. So for the group 2 populations, again, I'm showing here, this is severity, reaction class, and coefficient of infection. The reaction class is quite skewed towards susceptibility, but for the coefficient of infection, we see a fairly bimodal distribution, which may imply that a major QTL is conferring the phenotype. So we mapped these populations using composite interval mapping, and found a QTL in chromosome 5D and chromosome 7C in this population, and another 2 QTL 40.1, which was detected in the previous group. And that means that there's indeed a QTL in this region, and another one in chromosome 7A in this population. So for the remaining 2 populations, we only detected one additional QTL, and that is also in chromosome 4D, named 40.3. Again, just zoomed in version of the QTL that I have shown. This is the one in chromosome 5D. It has an effect of 32%. These are the CASP and PACE markers that I developed to select for that QTL, still high LD. And the same here for the QTL in 7C with an effect of 21%. For the CI7035 population, here's the QTL in 40.1 with an effect of 31%. This one is in 7A, has an effect of 20%, still high correlation on that significant region. And lastly, this is a QTL with a smaller effect, 13%, but we can still see high correlation between markers in that significant region. And that QTL is 6 MB away from the previous QTL that we have identified. So again, I looked at a representative QTL, the gene content of them. We see some resistance gene analogs here, and some other genes that are PIK2 that can be that have been identified in other crops such as rice, and they can be candidate genes for APR. Next, I'll go to the marker-assisted pyramiding of APR loci. So these are all the QTL that we have identified in this study. All 7 of them, 3 in 4D, 1 in 5D, here in 6C, 7C, and 7A. And there's one additional from Dr. Howard Rines. This is in chromosome 4A. So in total, we have 8 APR QTL available for pyramiding. And of course, the sizes of the chromosomes are from the 3098 reference genome, which is an estimated size of around 11 gig. And we did diallele mating design for pyramiding this QTL. I'm only showing you set 1 here, but we actually did 3 sets. We're still working on sets 2 and 3. But in here, we have 9 lines, and we did diallele mating design. So we crossed every parent with all other parents, and also reciprocal crosses. So we use 1 parent as a female and as a male. And we included here the 2 MNBT lines from Dr. Rines, and there are 4 total QTL that are available in these lines for pyramiding. And we started this in 2017, and we have advanced lines right now. This shows the segregation of the PACE markers that I use for selection. So I used 2 markers for selecting 2 flanking markers for selecting a QTL. So these 2 markers are for the QTL in 5D. These 2 are for the QTL in 7C. These 2 are for QTL in 4A, and these 2 are for 40.2 QTL. So I use a SNP haplotype analysis for selection. So they have to be homozygous resistant here and homozygous resistant also on the other SNP. And we can see good segregation of resistant and susceptible marker genotypes, and also the heterozygous genotypes. So I analyzed the effect of the number and type of QTL on the coefficient of infection, and found that as you increase the number of QTL, the coefficient of infection decreases. But we cannot differentiate the difference between 2 and 3 QTL, probably because of the sample size and also the replication. We only tested last year in the Bacter nursery. And I also looked at the effect of each individual QTL on the coefficient of infection, and found that QPC APR4A has the most significant effect compared to the other QTL. And this QTL was from the MNBT line. For 2 QTL combinations, The same is true. Whatever you pair with chromosome, with QPCA pair for a QTL, it will be significantly lower compared to any other QTL combination. And the same for 3 QTL combination. That's the maximum that we have obtained from set 1 of process. Anything with a 4A will be significantly lower. Here I'm showing you the phenotypes of the advanced lines that we have in the Bacter nursery last year. Here is a line with 3 APR loci. This one has 2 APR loci. This one has 1, and this one has 2 APR loci. Again, the type of the phenotype will depend on the type of QTL that the line possesses, but they're all pretty resistant in the Bacter nursery. Also, I looked at the effect of using the lines as male and female parent in the crosses. For most of them, it doesn't really matter if you use them as a male or a female, but then except for BT1021, when you use it as a female, the CI is significantly lower compared to the others. So it was really interesting, but this is just preliminary data. But it is interesting because in wheat, there's a gene called YR36, which I mentioned in the introduction. And upon signaling, that gene gets transcribed and translated, and the product goes into the chloroplast, and it induces a series of phosphorylation events, and that causes cell death. So there can be a favorable nucleus and cytoplasm combinations, and that will be worthwhile to explore in oat as well. And of course, we have to do fine mapping to really identify the causative gene for APR. But it is interesting to think about it. Lastly, I'll go through the GWAS study that we performed to detect effectors in crown rust. So as Oluseyi showed earlier, there's a drastic increase in virulence from the 1990 to 2015, and we wanted to know what caused that. And my job was to conduct the GWAS to identify putative effectors in the pathogen. And so this is the SNP calling pipeline for the GWAS. We call the SNPs against 2 reference genome, 12ST80 and 12NC29, and that led us to about 800,000 SNPs that we use for GWAS using MLM model in Tassel to account for the population structure and also the relatedness of the isolates. Here are some results of the GWAS. So I conducted the GWAS in both using both set of SNPs that were called against the 2 reference genome, and basically I detected the same peak. So these peaks in orange are orthologous to each other. So that one is for PC14, this one is PC50, PC51. So the purple one doesn't contain any predicted effector. Stainless and TAM0405, and that is consistent. The detection of single peak is consistent with a 1-AVER protein that is being detected by an R protein. And for TAM0405, there are more candidates because probably this differential line contain more resistance genes. And we have this group of differential lines, PC48, 52, 57, 61. We detected 2 peaks. These 2 peaks are orthologous to this. But then in the orange one, we have 2 candidate effectors. And those effectors may be detected by variants of a resistance gene in the differential line. In summary, we detected 7 QTL from 9 mapping populations, and we developed CASP and PACE markers for selection. And the reference genome enabled anchoring of the QTL to the physical map. And in the pyramiding, we combined up to 3 QTL, and these lines will be tested in different environments and will be released for use in breeding with the associated markers. So this will be publicly available. Also, fine mapping, cloning, and functional genomics are needed to really examine the mechanism of APR in oat. And on the side of the pathogen, we identified putative candidate APR effectors. And we also found that the drastic shift in virulence was caused by mutation, a combination of mutation and sexual recombination in the alternate host, which is buckthorn. And this information can be used in the future for diagnostics, tracking of pathogen movement, or even race typing based on genetic profiles. So with oat crown rust may be the worst or the ugliest disease of oat, but with all these recent advancements and together with the advancements of the other scientists all over the world, maybe we can make it not as bad of the disease in the future. I'd like to thank everyone in this slide, especially Shahryar and Roger for helping me with the APR mapping project, Marty and Howard Rines for the developing the populations and the MNBT lines. Jason actually did a calling and they did a SNP calling against the OT3098 reference genome, and a lot of the work on the pathogen side has been done in collaboration with Melania Figueroa. We'd like to thank our funding sources and thank you everyone for joining us today, and we're ready to take any questions that you may have. Thank you.
Thank you, Eric, Lucy, and Shahryar. A quick reminder to everyone to please use the Q&A function to submit questions, not the chat function. This is important for the recording so that the questions are preserved. So if you've put a question in the chat, please copy that and resubmit through the Q&A function. Thank you very much. I'm going to turn it over to Charlene.
Yes, thanks very much, Jim. Yes, thank you very much to our speakers. Those were wonderful presentations, and I'm really impressed at all the work that's been done and how far everything has come in recent years. So that's all very wonderful. Let's see. Yes, so we do have things in the chat and in the Q&A. I'm actually going to pop over to the chat quickly first, just because Steve Harrison had made a comment saying that he noticed PC35 was more effective in 2015 than previously. And Shahryar, you did answer everyone there and said that you had to look at sample size and isolates that were collected, and you're going to check to follow up on that comment, yes?
Yes, I was. And I was trying to do the picture right now to look at to see where the samples came from. Because, I mean, it could be that the only reason was some of the samples that were collected was not from the same region. So. Right.
Okay. Thank you.
A quick request to Eric, would you please stop sharing so we can all see the panels?
So while Eric is doing that, also over to the Q&A. So a couple of things have been answered, but we'll go over these again anyway. Jennifer Mitchell-Fetch was wondering how much infection was on the susceptible check. And again, Shahryar has said that Marvelous is the susceptible check and is always red. So wonderful name for a highly susceptible cultivar. And Craig Carlson said, nice talk. Do you have plans to do gene expression work in these populations as a supplement to fine mapping? And Shahryar has stated not at this time. So going to the open questions now. So from Steve Harrison, and actually looks like Shahryar is typing an answer, but maybe we'll read this one out anyway.
No, I'm leaving it up to Eric to answer. So.
Oh, okay. So QPC APR4A seems to be very effective and easy to screen for, particularly with one additional QTL. How common is this QTL in oat germplasm? And if not common, why has it not been selected and increased in frequency over time? Good question.
I think it was from Venus trigosa. So Dr. Howard Rines introgressed it from Avena strigosa to hexaploid oat. So I would think it is not very common.
That does make sense. Actually, I think I saw that Howard was on the line here or participating. Howard, if you did have anything to add at some point, you could put your hand up or ask to be made a panelist as well, because I mean, you've been working on all this sort of thing for many, many years. And so it's the foundation for a lot of this other work. In the meantime, are there any morphological markers for crown rust resistance? And that's from Anup Dakal.
If you're talking about crown rust resistance in the plant, I don't know of any. I mean, I haven't looked at any.
Yeah, I haven't heard of any either. Yeah, I don't think it's linked to any specific morphological marker, is it? Does anyone else know the answer to that question? Well, someone can say if they have an answer. So moving on to Paul Richter, how do the seedling resistant genes program cell death, and how does the APR program death confer better resistance management? Who would like to address that?
I'm leaving it up to Eric. It's his work.
What is the question? How does singers? So I think the mechanism of APR is not really completely understood. So in wheat, we have these three genes, LR34, LR67, and YR36. But from the proposed mechanism of YR36, it is different from the traditional receptor and ligand mechanism, which is in seedling resistance. But so YR36 product goes to the chloroplast, and the phosphorylation in there causes cell death compared to, so they're, I think this, my understanding is that they're being activated differently, but both can cause cell death. But of course, the race-specific resistance or seedling resistance will induce a stronger response compared to the APR. Because APR, we see necrosis, but there's still some pustule. So pustule and then necrosis around the pustule. Whereas in seedling resistance, you can either see, you may not see the hypersensitive response with the naked eye. So it's just very microscopic HR. So I don't know if that answers the question.
Okay. Just following up to the question about the morphological markers, Jim Menzies has said in the chat that he doesn't know of any either. So now Steve Harrison has another question. Could the PC35 avirulence have some fitness advantage?
Good question. We have to test. I have to look at the more complete data set, but my suspicion is from that graph that Oluseyi showed, that might have already been defeated. So
Yeah, it'll be interesting to see the answer to that question.
yep, but
Also,
I suspect not. So
people are recycling a lot of genes too, which is kind of a whole other topic to cover. Won't get into that maybe right at the moment because Catherine has a question here too. So Catherine Howarth says, you mentioned that you called your SNPs against the OT3098 sequence. Were there SNPs not found in the sequence? And how did the genetic maps of your populations compare to the physical sequence? Also, were you using version 1 or version 2, or or both at some point?
We call it against the version 1.
But my question is if Catherine is asking about the SNPs in the pathogen or SNPs in the plant, because the pathogen was called against the 2 genomes that were sequenced, the Rust genomes that were sequenced.
My guess would be the plant. Catherine, perhaps you could clarify that.
Yeah, I think it's the plant. It's OD3098.
Okay, the plant, okay.
I actually have shown the comparison of the genetic map and the physical map. So yeah, some of them can be, some of them, I mean, I have detected some recombination in the genetic map compared to the physical map. And some of the regions are just linear, just the same order as the physical map.
Yeah, and of course, OT38 is just one sequence too, right? So one particular line. So I suppose as we get more genomes, we'll find out more about these rearrangements. And actually that leads, I suppose, to a bit of a question. Where you're finding these genes, I know, for example, for a lot of the single genes, they're often found in areas of really reduced recombination and where you have a lot of sort of weird cytogenetic anomalies. So do you see that same sort of thing with these APR genes? Do they, especially if something's been introgressed from a wild species? Did you look at anything like that at all?
We didn't really look into that. Yeah.
I mean, you've already done an awful lot of work, so I suppose it's not fair to sort of go on about yeah. But just curious to know.
I guess Eric's answer is no.
Yeah, I guess that's fine. That's perfectly fine. Yeah, I was
see
also,
that,
sorry, go ahead, Jim.
oh, sorry, I didn't mean to interrupt. I see that Jennifer has raised her hand.
Oh.
Jennifer, I'm going to give you speaking permission. You should be all set, Jennifer.
Actually, it's not me, it's Tom that wants to ask the
Oh,
question.
hi, Tom.
So can you hear me?
Yes.
Okay. So I wanted to address Paul's question, and I guess I'll maybe phrase it with my experience in the stem rust world, and using, you know, APR versus major gene resistance is a hot topic and has been for many, many years. So if you look at different places, like CIMMYT will use an approach using APR and trying to stack those genes. Whereas, for instance, in Canada, we've typically used an approach to stack major genes, and particularly in wheat. Now for oats, the problem number one is that for major genes in crown rust, it's like, yeah, there's a pile of genes that have been named. Many of them are probably the same thing because they didn't do allelism tests. So that's one problem. And you know, in recent years, you know, since I started work, there hasn't been really a lot of work to go out and find more genes. Because yeah, they do seem to break down fast because of the buckthorn. In wheat stem rust, of course, we got rid of barberry, which is probably one of the biggest factors that's led to the success that we've had, right? Because if the pathogen, frankly, makes it very difficult to allow it to mutate or go through sexual recombination, you're going to gain an advantage because you can put out those R genes without getting changes in virulence as quick. With crown rust, of course, we got a big problem. But you know what? There are I mean, what I remember talking with James Chong and looking at stuff in nurseries that we put out and looking at wild species like Sterilis and diploid species, and Howard may chime in on this. There's lots and lots of R genes out there. It's a matter of finding them and then putting them in combination so that they don't lose effectiveness. So I guess the long answer or the short answer to this question is, you know, you can get effective APR resistance, which also can actually be race specific, by the way. But you can also get effective major gene resistance. The problem is that we lose it. And so you can't keep, and I've said this actually, I put this out in a recent paper that I just read. You can't keep putting out single genes alone. They're gonna keep going down. You gotta actually get somebody that's motivated enough to find some new genes, put them in tandem, whether it's APR or major, and then release it. Because if you put it out one at a time, you could very likely lose it. And it could even happen with an APR gene. We believe it's less likely, but we know if you put out, for instance, when SR2 was in there, it was good for a while, but if you get enough pressure from the rust, it will not hold. Not one gene, it won't hold. And that's why Simit's using, for instance, probably SR2, LR34, and some other genes to try to keep that thing effective. So anyway, that's my long diatribe, and maybe Paul Howard may have some comments as well. So I'll let it go for there.
Well, Howard's welcome to chime in if he wants to. In the meantime, yes, thanks, Tom. That's really great. You've made some really
To
good.
Tom's point, this is why we haven't released these APR genes as single genes for the public. So that's why we've been pyramiding. That was second part of Eric's work. And essentially the pyramided line will be released. And I'm working with USDA to have a material transfer agreement that would require, the only requirement would be that the pyramid has to be kept together. So we will release it to the public as long as they'll keep the pyramids together, because if somebody doesn't, then we're back to the ground zero. So
There we go. Yeah, that sounds like a very smart idea.
yeah, so you put the work into it, you got to make sure it stays that way.
That's also a very good point. That's great. And actually, I suppose this also ties into Mike McMullen's question, which he has up, which is it safe to assume APR will lead to durable resistance? So
I wonder if Eric, has this been proven or not yet?
Yeah, as Tom mentioned, some APR genes can also break down. So we have to know the mechanism of each of these resistance genes. And that's why further work on the fine mapping and maybe even cloning of some of the APR genes in oat is worthwhile, because that way we will learn how APR works in oat. And yeah, it's safer for now to just combine this APR, just in case one of them breaks down, so the other one can, salvage the crop.
yeah, for sure. It's a really important area of work. So it's
May I
wonderful.
ask a quick follow-up to that?
Oh, yeah, well, you're the pathologist, actually, Jim. So go ahead.
So just for reference for everyone on this call, I work really in potato late pathosystem. One of the concepts that's emerged in potato is monitoring the pathogen effector suite so that we can deploy our genes strategically to match what the pathogen population is doing. So Eric, is that a direction that oat crown rust is going in, and how far out is that?
Yeah, I think with the USDA, so we do, the USDA does surveys every year, right, for crown rust. And so with the crown rust research taking off, we're identifying effectors, we're doing chromosome level assemblies, and maybe we can do some markers for these effectors. So we collect these isolates from all over the US, and then maybe we genotype them with the markers. So we know what AVR genes are predominant in the population, maybe in the Midwest region or in the Southern region. And that will guide us on deploying resistance genes, because we know that this particular effector is being recognized by this gene or gene combination. So yeah, I think that's what I see in the future. Maybe we can use that information to guide the growers on what varieties are the best to plant based on the predominant crown rust population that is present in that region.
And this very much ties into what Oluseyi was saying. We need rust samples, and especially with the pandemic and restrictions on travel, more rust samples we have, better we are at predicting what's happening in the pathogen population.
Yeah, and actually Jennifer Mitchell-Fetch, or maybe it's Tom, has something else to say here. Durable resistance is only known when a line with the genes has been grown for many years over many environments. Apparently there's an original description by Roy Johnson. I'm not actually familiar with that reference, but yeah.
They've also raised their hand, and then I see Howard as well. So first Tom or Jennifer, and then
Sure,
we'll go to
yes.
Howard.
They can fight for it.
Go ahead, Tom or Jennifer, if you had additional comments.
Yeah, okay. Yeah, so I mean, that's another hot topic of discussion, which I won't go into as much diatribe, but many people have been look in the tech in publications, they'll talk about durable resistance, and it's all about APR. And I remember actually talking with Roy before he passed away, and he was quite incensed about that, because he said the mechanism has nothing to do about whether it's durable. Durable, the concept is it is effective over a long time in a wide host range. So it needs to be out there for many years. So a good example of a durable gene in barley is RPG1 for stem rust. And it's a major gene, but it's been effective for, my goodness, since 1937 or something. So, but yet certainly we think that, you know, because APR seems to be less prone to breaking down, that we call it durable. And certainly it seems to be like, well, if I combine more of those together, they should last longer, but it's hard to call it durable. So I don't know, maybe we need a new definition out there for that, because it can be a bit of a bone of contention about using a previously described thing by Roy for that purpose. So anyway, that's what I would answer on that.
Well, Tom, I'm maybe going to challenge you a bit here and say, if you would like to write up some sort of description of all of this that I could publish in the Out newsletter, then perhaps you would get the word out to the community that people really need to think carefully about how they're using these definitions and how they're thinking about some of these things. So anytime you'd like to do that, I would welcome that. Howard, did you want to make some comments now?
The gene that I worked on, I haven't been following it too closely since I've been retired for about 10 years now. But although I've been working on that gene since my retirement, but nothing in the last couple of years. But the problem, that's the gene that's most effective, the R gene by itself. And the problem with it is that it also imparts early necrosis. So it has a phenotype effect that has not been separated clearly from the resistance. And there has been selection, or there was selection or background effect. And there are other genes that have reduce the amount of or earliness of the early necrosis effect. during pre-harvest. And Kevin Smith has had students and postdocs who are working on keeping the gene that prevents quite such early necrosis together with the gene itself for resistance, and has been trying to put that together in lines so it hasn't and for reasons already given, the gene that gives the high resistance has not been released as a single gene because of the fact that it could then promote early mutation of that gene.
Right.
So people are working on putting it together with other genes.
Great, thank you, Howard. One thing, I've just put a couple of links in the chat actually, because we were also talking about, making the collections of the rust and so on. One is to the serial rust bulletins, and the other one is for the serial rust listserv mailing list, so that people can use those to contact Oluseyi and so on.
Yeah, thank you for doing that.
So they're both there. Also, there is.
Just one more thing.
Sorry,
And
go ahead.
just send an email to Oluseyi. She will send you a brochure to pass out. She will send you envelopes to send. It doesn't cost you anything. Just you have to collect the leaf, throw it in an envelope, and mail it to her. So.
But that's solely for the United States, right? The self-addressed envelope thing?
I think so.
Yeah, only
I
for the
wonder if
United
you can make a Canadian version too. I don't know if the mail is that much more expensive. So, I mean, it's just, I don't know. How much does it cost to mail an envelope from Canada to US?
Gosh.
40 cents, 50 cents?
It's a little bit more than our standard domestic mail, of course, but it's
Okay.
not, oh, my light just.
If
went
there
off.
is an interest, just let us know. I'm sure Oluseyi will figure it out for you.
Right, yeah.
I'm
Well,
volunteering it.
there are people here, of course, who collect the information and samples as well, but
Right.
I know, and Tom could actually speak to how that's going right now, perhaps. Oh, actually, Tom has just said here, no, we cannot send from Canada to the US. So everything should go to Jim Menzies for here in Canada. But I was also thinking of some other countries as well, because, well, as you know, Shahryar, we were talking a little while ago about somebody who was looking to have some things identified, and he's from way far away. So yeah.
We have the AFS permits and can receive international samples. There is a process for it to go through, and there is a different packaging process. So if people are interested in sending us samples, we'll be happy to just give me a email or send me a note, and we can talk about it. So.
Yeah, maybe you can write something up about that too, which we'll also advertise through the newsletter and then through, well, the serial lists and so on and so forth. So great. I don't see anything else, and I guess we are out of time, essentially. It's afternoon. Is there anything else anyone would like to say, add, comment, ask another question?
A big thank you to our panelists today. You did a great job, and thank you everybody for the great questions and engagement.
Mm-hmm. Should I put up the slide for next time, or?
Thank you.
I can do that.
Okay, there you go. Yes, speaking of oats, we'll continue. So the one next month will be Axel Diedrichsen. And Axel is Canada's point of contact for the International Treaty for Plant Genetic Resources for Food and Agriculture. And that's the sort of stuff he's going to be speaking about in his talk about the impact of global changes on oat conservation and exchange, specifically talking about plant gene resources of Canada, but also, I mean, it affects everyone worldwide. So certainly hope everyone can join us for that.
And we will announce registration for that very soon. So watch your email and the newsletter.
Mm-hmm.
All right, thanks again. Bye, everyone.
Thank you, everybody.